So first of all, thank you very much for the organizer for the invitation to present my work today. I'm going to talk about the therapeutic potential of cellular senescence. So to start with, what is cellular senescence? Cellular senescence is a stress response aimed at preventing the propagation of damaged cells in the organism, and it was described more than 60 years ago by Leonard Hayflick. Initially, we thought that cellular senescence was triggered by telomere shortening that happens naturally with age, and this is what nowadays we call replicative senescence. However, now we know that senescence can also be triggered by different types of stress, and that includes oncogenic activation, oxidative stress, mitochondrial dysfunction, irradiation, exposure to chemotherapeutic drugs, or even exposure to COVID-19. So what all senescent cells and all senescent types have in common is an irreversible cell growth arrest that is implemented thanks to the activation of the P53 and P16 pathways. Beyond the irreversible and stable cell growth arrest, the two other hallmarks of senescence are the acquisition of a senescence-associated secretory phenotype, or SASP, that is a complex mixture of chemokines and pro-inflammatory molecules that drives chronic inflammation, and also that senescent cells are intrinsically resistant to apoptosis. They are more resistant to apoptosis than non-senescent cells. So the framework, how we understand senescence now, is that basically in response to damage, when the damage is too acute, too severe, cells will likely undergo apoptosis. When the damage is light, cells will likely quickly repair themselves. So, but then there is this intermediate state that is what we call senescence, when in response to damage, cells will undergo this irreversible cell growth arrest, and then eventually, eventually, senescent cells will acquire this secretory phenotype that we call SASP, that is very important because it reinforces and keeps stable the cell growth arrest. It is also important because it helps to propagate the senescence phenotype to neighboring cells, and this is what we call paracrine senescence. But also the secretory phenotype of senescent cells is important because it recruits the immune system, and then the immune system, macrophages mainly, will phagocytize the senescent cells, so the tissue will be regenerated. And this is the chain of events that we think happen during the physiological roles of senescence that it has during normal development, wound healing, or tumor suppression. However, especially with advanced age, there is a net accumulation of senescent cells, and this is for two main reasons. One is because with age, there is more damage. There is more DNA damage and all types of damage. And second, because our immune system is less efficient with age to eliminate senescent cells. So as a result, this accumulation of senescent cells in the tissue doesn't allow the tissue to repair and regenerate, so that leads to tissue dysfunction. But also importantly, as senescent cells have this secretory phenotype that is pro-inflammation, this accumulation of senescent cells will also drive chronic inflammation with effects in age-related pathologies and cancer. So because of this, many of us believe that senescence can be considered the central hallmark of aging, because if we pay attention to all the hallmarks of aging, senescence is such a phenotype that integrates all of them. For instance, epigenetic alterations, telomere erosion, DNA damage, or mitochondrial dysfunction, they are all well-known triggers of senescence. But also, senescent cells lose the normal protein homeostasis, they show metabolic alterations, and also importantly, senescence drives aging, because the main outcomes of aging are the exhaustion of the stem cell pool and also chronic inflammation, and senescence drives both of them. But an important point that I want to highlight is that senescent cells are not just mere bystanders of the aging process. It's not that they just accumulate with age. They accumulate with age, but they also play a causal role during aging, because we know this thanks to two excellent piece of work by the labs of Jan van Dersen and Darren Baker, where they prove using transgenic mice that the selective elimination of senescent cells increases health span and delays age-related deterioration. So, of course, this work came as a revolution in the field, And it started a race to identify what we call senolytics, that are these compounds that specifically kill senescent cells. So initially, we were looking for compounds that were able to target the apoptotic resistance of senescent cells, with the idea that during advanced age, we can still eliminate the senescent cells and allow the tissue to regenerate. And we have discovered plenty of senolytics so far that are great in preclinical models, but unfortunately, most of them have severe side effects that prevent their move to clinical trials. So therefore, this is a very promising area of research, but there is still the need to identify better senolytic compounds. So in this context, we started this work, and our aim was to identify novel senolytic compounds by doing high-throughput drug screenings. Our model was primary cultures of human embryonic lung fibroblasts that are called IMR90, and these IMR90 have been modified to express a cassette that has a protein that is the estrogen receptor fused to an oncogenic RAS. So we have these primary cell cultures, and if we add tamoxifen to the media, the cells undergo senescence. So in using these cells as a model, we tested the senolytic potential of the LOPAC library that includes more than 1,000 different compounds. So this is the design of the experiment. Basically, we plate the cells. The day after, we add tamoxifen to trigger senescence. We wait more or less 1 week, and after 1 week, we add this big drug library to see if any of these compounds could have any senolytic potential, looking at the cell survival of the cells. And what we found is that there is a small group of compounds that actually are able to specifically kill senescent cells without affecting the viability of the non-senescent cells. But we didn't want to just identify a senolytic with potential to treat oncogenic induced senescent cells. We wanted to look for a senolytic with a broad spectrum capabilities that was able to target different types of senescent cells. So to achieve this, we repeat the same high throughput drug screen, but in a model of therapy-induced senescence. This is the trigger of senescence in this case is the addition of chemotherapeutic compounds. In this case was etoposide. And then we merge the result from the oncogenic induced senescence screen with the therapy-induced senescence screen, and we came out with a smaller subset of compounds with senolytic potential. Among them, we selected oavine because it was the one with the highest potency at a lower dose, but also because we were able to demonstrate that oavine was also senolytic in the context of replicative senescence, doxorubicin-induced senescence, and palbociclib-induced senescence. So we really believe that this is a broad spectrum senolytic and highly specific. Then we wanted to see how this compound was eliminating senescent cells, and what we found is that it was doing so by triggering apoptosis. So what you can see here is a time-dependent increase of caspase-3/7 activity, which is a readout of apoptosis. And in fact, if we treat our cells with this compound that is called QVD, this is a pan-caspase inhibitor. So if we prevent the activation of the caspase, so if we prevent the apoptotic pathway, the senolytic activity of different cardiac glycosides is prevented, because this is also another important point. It's not just that the oavine has senolytic potential. In our knowledge, all the cardiac glycosides, so oavine belongs to the cardiac glycosides family, so all the cardiac glycosides that we have tested have senolytic potential. So it's a family of compounds with senolytic potential. And then we wanted to clarify how oavine was triggering apoptosis. So to do that, we did RNA-seq, and we combined the results of the RNA-seq looking at the proteins that anti-apoptotic and pro-apoptotic proteins, the ones that go up and down, and we combined the results from the RNA-seq with an SI and shRNA screen, knocking down all these proteins and trying to look which one was the relevant one to explain the phenotype. And what we found is that when we knock down NOXA, either with siRNAs or with shRNAs, oavine is not senolytic anymore, means that oavine triggers apoptosis of senescent cells through induction of NOXA. And then, of course, we assess the senolytic potential of these compounds in different in vivo settings, but today I will just show you a summary of the results that were obtained when we add these senolytic compounds into naturally aged mice. So we basically had a cohort of 22 to 24-month-old mice, and we randomly allocated these mice into vehicle or oavine. We keep the treatment in place for 12 weeks, and we were treating IP with oavine every 3 weeks. And then at the end of the experiment, we did, of course, different tests. One of them, rota-rot test, assessed motor performance and coordination, and we found that thanks to the senolytic treatment, these mice, these naturally aged mice, perform way better. They have better motor performance and better coordination. So senolytic is reverting, in a way, the age-related deterioration in these mice. And important for us, we found that this correlation of this relief of the aging phenotypes correlates with a decrease on the senescence markers, because as you can see here, the old mice treated with oavine show lower senescence-associated beta-galactosidase activity. That is one of the most common senescence markers. And to go a little bit beyond, we also did RNA-seq in the livers of these aged mice, and we confirmed that there is a decrease in the senescence signature in the liver of these mice, and there is a decrease in inflammation. So overall, what we show here is that cardioglycosides trigger apoptosis of senescent cells, that this oavine-induced apoptosis is mediated at least in part by induction of the pro-apoptotic BCL2 family member NOXA, and also that oavine is able to decrease the senescence burden in all mice while improving physical fitness and decreasing inflammation. But we didn't want to stop here, because we really wanted to explore the therapeutic potential of senescence. So we know there are other people working in SAS modulation with a type of compounds that are called xenomorphics. We explored the senolytic path, but then we also thought about trying to find drugs that could prevent senescence and explore what could happen, how these could be helpful in an aging context. So basically, we follow a similar approach to what we did before. We also use this primary human lung embryonic fibroblast, IMR90, with the cassette to trigger oncogene-induced senescence, and also with a different cassette to trigger replicative senescence, because again, we want to find compounds that have broad spectrum activity, not just focus on a specific type of senescence. And this time, we assess more than 1,500 drugs. So the design of the experiment is slightly different, because in this case, what we are doing is we plate the cells on one day, and the day after, we add at the same time tamoxifen, which is the trigger of senescence, plus the drugs that we want to evaluate if they are able to bypass or to prevent senescence. And in this case, we were using TGF-beta inhibitor as a positive control. So as you can see here, these are growing normal cells that they are incorporating BRDU at the same time that they replicate. And you see that if you add tamoxifen, the cells undergo senescence, so there is less BRDU incorporation. And if you treat with this type of compounds that are able to prevent senescence, you see that cells are able to still incorporate BRDU to still grow. So this is an example of a compound that prevents senescence. So after running this screen in both models of senescence, after doing several rounds of validation, we came out that our main target, our top hit, was 3DA. And then we did, of course, additional validation to prove that 3DA was able to prevent senescence, oncogene-induced senescence. So as you can see here, there is a dose-dependent increase in BRDU incorporation. I'm sorry. And then this is another way of looking at proliferation. This is called crystal violet staining. So as you can see here, if you leave the cells growing for 10 days, the plate is saturated, means that the cells have grown a lot. If you treat with tamoxifen, most of the cells will undergo senescence, so will stop proliferating. So the density of cells in this plate is lower. But then if we add 3DA, cells don't undergo senescence, they keep proliferation, so we recover the normal density of cells. And we also found that treatment with 3DA decreases the intensity of the senescence-associated beta-galactosidase activity that we normally see during senescence. And of course, importantly, we repeat the same type of experiments in replicative senescence, and it was also true. And what is 3DA? What does 3DA do? So 3DA is an inhibitor of AHCY, and AHCY is a highly conserved enzyme that is involved in the recycling of this compound, SAH, which is a byproduct of some dependent methyltransferase reaction. So basically what happens is that if you inhibit the activity of this enzyme, AHCY, with 3DA, there is no recycling of SAH. So as a result, this accumulates in the media, and the accumulation of SAH acts as a negative signal for methyltransferase reaction. So some dependent methyltransferase reaction stop from happening. So in a way, 3DA is an indirect inhibitor of some dependent methyltransferase reaction. So we wanted to prove if 3DA was acting on target or not. So if the well-known target of 3DA is AHCY, we thought, what will happen if we knock down AHCY? Will we see the same phenotype that we see when we treat with 3DA? Sorry. So what we did is, using short herpes RNAs against AHCY, we knocked down the expression of this gene, and basically we got exactly the same result that we were seeing when treated the cells with 3DA. So knocking down AHCY also prevents the stable cell growth arrest that we see during senescence, and cells keep proliferating. So basically this is a strong indicator that our drug 3DA is acting on target. And again, we did also these experiments in a model of replicative senescence, and the same was true. So to understand better the mechanism of action of 3DA, we decided to do RNA-seq, comparing the transcriptome profile of normal growing happy cells with senescent cells, and then with senescent cells treated with this 3DA, that is this drug that targets AHCY, and also with senescent cells that have the knockdown of AHCY, and see how they look alike. And basically we found that there was no difference between the 3DA treated cells or the cells treated with the shRNA against AHCY. In both cases, we see a clear downregulation of the senescent signature and of the SASP, the secretory phenotype of senescent cells signature, which means that AHCY inhibition prevents senescence, and one of the ways that it does so is through the inhibition of the SASP. So how exactly we go from AHCY inhibition to the prevention of senescence? So we know that when we don't have activity of AHCY, we block the activity of certain sun-dependent methyltransferase reactions, but we didn't know which one of them. So of course we look into many of them, and we found very consistently that treatment with 3DA prevents the accumulation of the trimethylation of lysine 36. And this is another way of seeing it by immunofluorescence. So you see that upon addition of tamoxifen, cells undergo senescence, and one of the things that happens is that most of the cells increase their levels of trimethylated K36. However, if you add 3DA and tamoxifen, cells do not accumulate this lysine 36 trimethylated. And then we also performed ChIP-seq of trimethylated lysine 36, and what we found is that there is, so this trimethylated lysine 36 is found in genes that are being actively transcribed. So what we found is that when we merge our RNA-seq data with our ChIP-seq data, what we found is that there is a very clear correlation between the upregulation of certain SASP genes and an increase in the deposition of trimethylated lysine 36 in those genes. So it seems that at least certain SASP factors need the deposition of trimethylated lysine 36 to be upregulated. And among the SASP genes that seems to be regulated by 3DA, we found very well-known SASP markers like IL-1 beta, IL-1 receptor, or NF-kappa beta. But of course, this is great, but we need to see how this could be relevant, in what context preventing senescence could be relevant. So the first idea that came to our mind was geriatric muscle stem cells, because there is a work like that we know for at least the last 10 years, that we know that normally in young people, when there is any damage to your muscle fibers, the muscle stem cells that are quiescent, they get quickly activated, they proliferate, and they repair the damage. However, with advanced age, these quiescent muscle stem cells become senescent. Instead of quiescent, they turn senescent. Therefore, if there is any damage to the muscle, they are not able to activate and proliferate and repair the damage. So the damage cannot be repaired. And this is one of the reasons why old people have sarcopenia, that is this weakness, this muscular weakness. And the reason is because muscle stem cells switch from quiescent to senescence, P16 goes up, and the regenerative capacity of the muscle goes down. So we decided to test 3DA in this context. And first, what we did is isolate muscle stem cells from young mice or from geriatric mice. We call geriatric mice that are almost 30 months of age, so are extremely old mice. So we isolated muscle stem cells from both young versus geriatric, and we keep them in culture for 1 week, adding every day a dose of 3DA. And what we found at the end of the week is that the senescence markers in those populations of muscle stem cells were down. So it seems that 3DA was reverting the senescent phenotype in geriatric muscle stem cells. But even more importantly, what we did is then take these cells that have been treated with 3DA ex vivo, we took them back into mice. So what we did is did a pre-injury in a muscle of the leg of the mice, and then we engraft muscle stem cells that have been treated with 3DA or with vehicle. And what we found is that, of course, young muscle stem cells are very able to engraft and proliferate and repair the damage, whereas geriatric cells are not able to engraft, or even when they are able to engraft, they are not able to grow and regenerate the damage. However, if geriatric muscle stem cells have been pre-treated for a week with 3DA, we saw a very clear increase in their engraftment capability and in their proliferation and regenerative potential. So it seems that 3DA in this context could be highly useful. So in summary, we have found that 3DA treatment prevents oncogene and replicative senescence, and we believe that this is due to on-target inhibition of AHCY. De novo methylation of H3K36 contributes to senescent initiation, and then ex vivo treatment with 3DA rejuvenates geriatric muscle stem cells. So this is all for today. Thank you very much for your attention. I want to thank the lab of Jesus Gil, that is where I did most of the job that I presented here today, and also the lab of Professor Mercè Pallàs in the Universitat de Barcelona, because they are hosting me I have recently joined the University of Barcelona as a junior PI, and they have been very welcoming and supporting, and they are helping me to settle. Thank you very much.